for me, one of the aspects of evolution that was very compelling was biogeography and the way different animals evolved in different environments. And you could kind of trace these back over geography. And that to me was very telling, if there was like this intelligent design, miraculous creation sporadically around the world, that wouldn't be an unexpected pattern. So tell us some of the factors for you that were really compelling to promote evolution that do not fit well with an intelligent designer or spontaneous creation type of a viewpoint.
Yeah, well, I think you touched upon one of the biggest ones. I mean, Jerry Coyne, who's a famous, biologist, he says essentially that, biogeography is such powerful evidence for evolution that he said he hasn't seen any creationist lecture, any creationist book, or any creationist literature that's even discussed biogeography. They just pretend it doesn't exist because it's so, such persuasive evidence. And yeah, you think, why did great polar bears in the Arctic, and there's an identical habitat in Antarctica where they don't live? Why are penguins in the Antarctic and not the Arctic, even though it's an identical environment? Why do you have cacti in the Americas, but somehow in the identical environment in the Middle East, right in Noah's backyard, they never got established? Why does Madagascar have 100 species of lemur and there's no other species of lemur anywhere else on Earth? Why does Australia have all the marsupials except for, a few opossums in the Americas? Why do islands not really have any native salamanders, amphibians, fishes, freshwater fishes rather? And so, a lot of these facts don't seem to fit with, this creation model of either independent centers of creation or dispersal from, Mount Ararat or wherever the ark landed. Much better, evolutionary biology model where, you have dispersal and vicariance and these things that have such explanatory scope and power to explain why we have this distribution of animals the way we do. Even Darwin noticed this. Darwin was, in the Galapagos, he found these giant tortoises up to 6 foot long, size of almost the size of a Volkswagen Beetle. And, he saw the Chaco tortoise on the South American mainland. And obviously he hypothesized that, they dispersed over there, rafted on there in some vegetation. They colonized these islands. And even on the islands, these tortoises are all huge, but they have different shaped shells depending on the selective pressures on each island. He found fossils of glyptodonts, which are, these giant shelled creatures that are, prehistoric. And he found, modern armadillos, in the same spot he found the fossils of the ancient glyptodonts. So they look very similar, but they're different in many ways. And so, biogeography, I think, is something that even impressed Darwin, and he wrote a whole chapter on it. So I think that is persuasive evidence for evolution. I would say if I was pressed to pick maybe one or two pieces of evidence, I think one to me that stands out is human chromosome 2. So, if we do share common ancestry with the great apes, so, science tells us that our closest relatives are the chimpanzees, followed by the gorillas and the orangutans. Well, there's a problem, or at least on the surface there seems to be a problem. The great apes all have 24 chromosome pairs. Humans have 23 chromosome pairs. And I would say that presents a problem because it seems very unlikely that we would have lost an entire chromosome during our evolution and we would have still survived as a species. And it seems equally improbable that orangs, chimps, and gorillas would have independently evolved an extra chromosome. That would seem to be the end of them as well. But then, in 2005, we get a peek inside the human genome and we have, all the great ape genome sequenced. And we look at our chromosome 2, and what do we find? Well, on a normal chromosome, you have telomeres on the end. So the ends of the chromosome have these very repetitive, specific sequences of DNA that are like caps on the chromosome. You only find them on the ends. We also have this region of DNA in the middle of the chromosome called a centromere. And so that's, important for cell division events. Put them in the center. So, here's a testable hypothesis, right? If this chromosome fused, we had 24, a chromosome fused, now we have 23, we should be able to look at that chromosome and see telomeres in the middle and centromeres not quite in the middle, but maybe a third and 2/3 of the way down. Well, that's exactly what we see. We see a chromosome, an extra long chromosome, right? Twice the length of chimpanzee chromosome 2A and 2B. And in the middle of the chromosome, we see telomeres. We see end caps in the middle of the chromosome. And then instead of having one centromere in the middle, we have one centromere about a third of the way down and another centromere about 2/3 of the way down. And so I'm hard-pressed to find any explanation rather than common assent for why we would possibly have a fused chimpanzee genome, or chimpanzee chromosome essentially, in our genome. So that to me is really powerful.